pleasure to introduce Ricardo Kriebel. He's a Costa Rican biologist who's passionate about exploring and conserving plant diversity. He got a master's degree in ecology and systematic biology from San Francisco State University, PhD from City University of New York, and did postdoctoral research at the University of Wisconsin-Madison. He is currently an assistant curator and McAllister Chair of Botany at the California Academy of Sciences. And I want to say, because I don't know if you hear this, Ricardo, how thrilled we all are to have this position filled at Cal Academy. It's such an important organization for California and Western botany. And it's just terrific to have you there and to have that position filled, a real chair. His research centers on the taxonomy and evolution of the princess flower family, Melastomataceae. My good friend Frank Almeida must be just delirious about that, and the diverse genus Salvia. So his title is Moving Flowers, Evolution of Floral Form, Function, and Pollination in Salvia. Welcome, Ricardo, and you can share your screen. Yes?
So thanks so much, Nancy, for that great introduction, and thanks so much for having me. And thanks to Andy Cabrera also for organizing the posts and all those interesting posts he made. And as Nancy said, my name is Ricardo Kriebel, and I'm a curator at Cal Academy right now. And I'm going to talk about moving flowers, evolution of floral form, function, and pollination in Salvia. And before I get into the sages, I just want to mention that, as Nancy said, I've been working on Melastomataceae for about 25 years. And I want to just let everybody know that I keep working on Melastomataceae, and I keep working with Dr. Frank Almeida. He still comes in a lot, and we're working on papers just right now. And part of my interest in the Melastomataceae is to continue making data available for researchers. So last year we made available this paper that provides a lot of data that people can use to test their hypotheses. And I also have been working on another database that's not out yet, but it is what I call Melastoma Outlines. And it's a database of outlines that can be used also for morphometric studies to improve taxonomy and look at the evolution of different traits. But today I want to talk about work I did while I was a postdoc. I was introduced, I knew some salvia, but I didn't know how fascinating they could be. And one of the fascinating aspects that I learned about salvia that kind of got me hooked on them was that their flowers could move. And so here is the Salvia forscale, and I'm going to pretend to be a pollinator with a tweezer. And as I approach the flower and try to get nectar, the stamens lever down. And this is what we call a staminal lever mechanism. And this is very common in salvia. Most salvia have this, but it's uncommon in the rest of angiosperms. Here's another example, just so you can see how fast this lever can be. And these levers allow salvia to place pollen very accurately on pollinators, whether it's the abdomen or the thorax, or the face or the back of the bee's head. And not only bee pollinated taxa lever, I want to show you this is a sunbird pollinated species at San Francisco Botanical Garden. And it also levers. And here's a hummingbird species at UC Botanical Garden. And it also levers. And I was lucky there to be able to see a pollinator visiting one of these salvia and activating the lever in the process. So you could see how precise sages can place pollen on their pollinators. So today I want to keep talking about moving flowers, I'll get more into detail about the staminal lever mechanism in salvia. And that's going to be most of the talk, aspects related to the staminal lever. But I'll also introduce what we're calling a petal lever mechanism in a few California salvia. And lastly, a third way that salvia flowers move is during protandry. Most salvia change sexual expression through time. They're first male, then female. But I'm going to show you a discovery we've made where we think there's one group of salvia that have stopped doing this. Now, as I go through the talk, I'll also share some details about the taxonomy and biogeography of salvia, some results we've obtained. I'll talk about the Salvia NSF project that we are working on. And I won't only talk about the lever, I'll talk about also the evolution of the corolla, the style, and the stigma shape, and how that relates to the lever. Then I'll try to answer a couple of questions today in this talk. When did hummingbird pollination first evolve in American sages? And also, why are there different kinds of bee flowers, bee-pollinated flowers in salvia? And lastly, I'll talk about a small study we did trying to understand the evolution of further complexity of the lever, in this case, these teeth that have evolved on some levers. Now, just to place us a little bit in the phylogeny of the mint family, this is a phylogeny by Rose and colleagues. And basically, Lamiaceae, the mint family, has 12 subfamilies. And the largest one on top is Nepetoides. And Nepetoides has 3 tribes, Menthe, Eschscholzii, and Ocimi. And within Menthe is the subtribe Salvini, which contains 3 genera: Salvia, Lepecinia, and Melissa. Lepecinia and Melissa are sisters, and they in turn are sisters to Salvia. And this leads us to how this staminal lever mechanism looks like. And so here's a Lepecinia, sister to Salvia, and you can see the flower to the left and the stamen to the right. It's a normal looking stamen with a long filament. And if I put that stamen next to a garden sage, Salvia officinalis, you can see that both have a filament and both have connective tissue, but that tissue is very prolonged in salvia, it's expanded, and that's what forms the staminal lever. And it is very reduced in Lepecinia. Now, both have 2 anther theca, but in many salvia, the lower theca can get aborted and form these really long staminal levers. In this case, these taxa have flowers in which the lever is blocking totally the access to the nectar. And you really have to push through there to get access to the nectar, always forcing the lever to function. Now, the lever has actually been known for a long time, since 1793. This is perhaps the first book on pollination biology by Christian Sprengel. And if you zoom into the bottom of the cover of the book, you'll see a salvia with an active lever. And if you go inside the book, you'll find further documentation and illustration of how the salvia lever works. So this is the first instance that we know of documentation of the lever. But there's been other really interesting studies, like this one in 1933, that showed or hypothesized that the stamens evolved first, the 2 anther theca offset, and then in each part of the world then became longer levers, but lost the lower theca, aborted the lower theca, and then further complications of the lever below with teeth and different kinds of paddles on the lever. But then molecular phylogenetics came along, and in 2004, this study by Walker and colleagues found that salvia was not monophyletic. In this study, the authors found 3 genera intermingled with good support among salvia. Those were the genera Doristecas, Perovskia, and Rosmarinus, all very small and lacking a lever, and that's why they were in salvia. Then in 2007, Walker and Seitzma revisited the phylogeny of salvia, added more taxa, added more gene regions, and then they found that there were 5 genera nested in salvia. And these are the 5 genera that kept being nested in Salvia. And what I want to note in this slide is that the colorful genera without a lever came as sister in this study to clades with a lever. So the authors suggested that the lever had likely evolved at least 3 times. Now, in 2017, more recently, we revisited this question with nuclear data, and we found the same result. We had data for 4 of those genera, and they were way nested in Salvia. So at this point, the taxonomist of Salvia in the team went ahead and made the combinations that were necessary, and then we started to also just reuse names that were available, like Salvia rosmarinus. And that leaves us with 11 clades of Salvia. Salvia has 2 stamens, and in that it's different than its sister group that has 4 stamens. And then here are 3 of the hypothetical evolutions of the lever. Now, if we take the 11 clades of Salvia and turn the triangles into the relative size of that subgenus, these clades are now considered subgenera. So Rosmarinus is subgenus Rosmarinus. And if we make these relative size, we find that subgenus Calosfaci with 600 species is by far the largest subgenus, and it's one of our focus. Why is this clade so diverse? Now, I want to briefly introduce you to these 11 clades because they all have something interesting. First, we have here Perovskia. Subgenus Perovskia is what people call Russian sage. Sometimes I found it to be commonly used as ornamental in Madison, Wisconsin, for example. And one of the fascinating things of Perovskia, subgenus Perovskia, is the presence of heterostyle. Here above is a flower with long stamens and short style, and below long style and short stamens. This is the only group in Salvia with heterostyle, except for a species in the California group, Salvia brandigii. But heterostyle is very rare in the Lamiaceae. So Perovskia is very interesting that way. And it has a very normal looking stamen. Now, rosemary is interesting because its stamen has a little projection, and it has been interpreted as the placement of a second anther theca that was aborted. And the extension between them is the connective tissue. So the question still remains, where did this prolongation come from? Could it be that it was retained after losing a lever? Now, subgenus Heterosfaci is the only one in North America and also in the Old World, and it can have stamens with 2 fertile theca or 1. Subgenus Salvia, with about 70 species, usually has 2 fertile theca. Sclarisages are really distinctive because the upper corolla lobe or lip is very curved. The 2 lobes, the gallia is very curved, and it also, with the lower lip, forms these kind of handlebars that the bees grab onto. And this upper lobe that is very curved, this upper lip of the corolla, is where this really long connective is housed. This is the lever of sclarisages. And then we get to Asia. This is subgenus Glutinaria. There's a few Dioicia species here, so it's interesting among other things because of that. And they can have 1 fertile theca or 2. Now, smaller clades here, still in the old world, Zumeria, 30 species, can have a small lever and usually does a few species very reduced. And then 2 sister subgenera, Doristecas with small white flowers, used to be a genus, barely prolonged connective, and Mariandra with small white flowers and barely prolonged connective. Finally, we get to America, where we have the absolutely fascinating subgenus Aldibertia, one of the reasons I'm so happy to live in California. They're so diverse in many ways, and the flowers are part of it. For example, look at the stamens of subgenus Aldibertia. Salvia californica has 2 fertile theca, and the others usually have, some have 2, but most have 1, and different prolongations of that connective. Lastly, we have subgenus Calosfaci from mostly Mexico to South America and the Caribbean, a few species in the United States. And this group has many interesting things. Right now, I'm going to fast forward the connectives being long with projections. and frequently straight, which we hypothesize is due to hummingbird pollination in a lot of them that matches better their straight rigid beaks or straightish rigid beaks. Now, at the same time that all this systematic work was being done, all these phylogenies, there was a lot of work being done on the pollination biology of salvia. And I want to highlight the work by Dr. Regine Klassen-Bockhoff and Dr. Petra Vester, who was Regine's student. They studied the pollination of a lot of salvia, and also in one of their studies provided the list of all the salvia and what was the likely pollinator of all salvia. And they provided different ways to see this. Here are the 6 centers of diversity of salvia. And what they pointed out is that in the old world, they're bee pollinated as far as we know. And there's only 3 in South Africa that are sunbird pollinated. But in the New World, there's about 200 hummingbird pollinated salvia. So that's a big difference. And this information was used already in really neat studies like this one by Itzi Fragoso and colleagues. This interesting study built the phylogeny of subgenus Calophaceae and then mapped the pollination syndromes on the tree. They found that the ancestor there in the middle was likely bee, and hummingbird pollination evolved at least 13 times. So we wanted to revisit this with the phylogeny of the whole genus. And that's when I ended up in Wisconsin around that time. And Professor Ken Seitzma said, would you want to be part of this collaborative research? Help us in some aspects of this grant. And I said, of course, this sounds really interesting. I, at that point, was learning about salvia. And this was together with his former student, Dr. Brian Drew, who's an expert in Lepidoptera and in Lamiaceae. So we worked on this grant. The first year we didn't get it, but the second year we got it. And 2 of the main things we proposed was to start the next generation sequencing phylogeny of salvia. And the second one was to generate a morphological data set that was going to be based on species plates or morphological plates. The idea was we go to the field, we collect the plants, and some of them we dissect and we put in a flatbed scanner. We scan the plants, and with that, we produce plates that can be used to illustrate that species for identification purposes, or we can use these plates to extract morphological data, such as outlines for morphometric analyses that can then be matched to the phylogeny to test hypotheses and then make the data available. So we started to do that, and here are 6 of those plates. We tried to have at least a leaf in adaxial and one in a abaxial surface for every species, a corolla with and without the calyx, stamens, and styles. Now I want to highlight in the bottom right is Salvia piscidica here with compound leaves. You can see it has digitally compound leaves, which is really unusual in the family Lamiaceae. Here's 6 more plates. If you see the top left, you can see a salvia that when it's in the calyx, it looks basically black. And then to the right of that one, I want to highlight Salvia cruikshanki with a tuft of hairs below the stigma. You can see in the top bottom right of that plate. So it's a spoiler because I'll talk more about that, but only subgenus Calophaceae, as far as we know, has evolved hairs in the stigma, below the stigma. Okay, and I'll talk more about that in a few minutes. So then we teamed up with some folks from Florida State University that are good at doing the anchored hybrid enrichment approach, and we generated a data set originally with 35 species and outgroups for 316 nuclear genes. We built a tree that looks a lot like what we were getting before. And then we used this tree as a constraint to add to the matrix the rest of salvias available in GenBank for nuclear ribosomal DNA. That allowed us to create a phylogeny with 528 species, 519 are salvia, 7 lepidoptera, and 2 melissa. And with this dating analysis, we find that salvia is about 32 million years old, and we scored them for biogeographical region. And what I want to rescue from this slide mainly is towards the base of the tree, you can see the second node in is the common ancestor of salvia, and it's very turquoise. So there's a high probability salvia originated in Southwest Asia. And the two dispersals that I want to mention today here is a dispersal to America about 22, 24 million years ago. That gave rise to the California clade, sister to the Mexico to South American clade. And a second dispersal to North America about 10 million years ago gave rise to the small clade of southern United States, a very interesting small clade there. Okay, so we had a background about the biogeography and the phylogeny of Salvia, and now we went ahead and scored all the species for pollinator to revisit and try to understand how many times hummingbird pollination may have evolved, especially in subgenus Calophaceae, where most of them are by far. And when we did that reconstruction, we found a surprise. Here on top is subgenus Calophaceae, and we find that the common ancestor of Calophaceae was likely hummingbird pollinated. And then subsequently, there were many losses of hummingbird pollination. And that was a surprise because previous studies had suggested hummingbird pollination evolved many times, but when we included the whole genus, this is what we obtained. And then we said, okay, let's see what the morphology suggests. If a hummingbird pollination evolved early on, we might find changes in morphology that match that. If hummingbird pollination evolved very recently, we might find several changes very recently in morphology. So we went to our plates and we first did the corolla. And what we did is we took the corollas and we made outlines of them. And with those outlines, we can use this approach called elliptic Fourier analysis, using the R package Momox that decomposes the outlines into a series of ellipses, extracts these coefficients that then you can summarize using principal components analysis and generate a morphospace. Here's a plot of all the corollas, and to the right, you see the very bilobed corollas like sclarisages. And to the left, you see the very narrowly at the mouth lobed corollas. Those are more hummingbird sort of species. So we're going to take this data now, and we're going to match it to our molecular phylogeny, and we're going to use a shift analysis with this package called L10U, and we're going to ask, is there a shift in morphology that matches some of these changes in pollinator? What we find is one shift in the sclarisages for the flower. So we did not find here a clear shift to hummingbird pollination in subgenus Calophaceae. Now, these hatched lines that I've added are the confidence interval for the crown radiation of hummingbirds, just to show that when hummingbird pollination evolved, yes, hummingbirds were around, okay? Because that's important to point out. Now, if we include two axes of morphology of the corolla, we do detect one of the shifts back in Calophaceae to bee pollination. So flowers with very long lower lips are detected. So there is some signal here about the reversals at least. So then we went to the levers. These are more direct touch with the pollinator, and they're easier to quantify. They're simpler morphology. We make outlines of them. We calculate a morphospace, and we find in the morphospace that in the x-axis, you get the straight levers that we hypothesize are related to hummingbird pollination versus the curved levers to the right of the morphospace. We're going to match that axis to the phylogeny, and we're going to ask, is there a shift concordant with an early evolution of hummingbird pollination or more recent? And what we find is that about 20 million years ago, in these magenta lines, we have a shift to straight anther connectives. And if we overlay pollination, you can see that that is about the time that there's strong support for the evolution of hummingbird pollination. We then went, and by the way, then they stayed similar even though the bee species evolved within that clade. And then there's the styles. We suggested the same. There's very straight styles in Calophaceae. So we went ahead and did the morphometrics of the style, generated a morphospace. Again, the main axis is straight to curved. We match that to the phylogeny, and we find two deep shifts, two main shifts, one in the sclarisages to very curved styles, which we had obtained before. So it kind of makes sense to match the corolla shape. But then we also get to straight styles in Calophaceae, And if we overlay pollination, that is about the time that we estimate hummingbird pollination. There's less taxa for styles, so it looks smaller, that clade, because we had less. So we're getting these changes that are about 20 million years ago, and there's one more character, the stigma, that was suggested to perhaps have evolved similarly. So we wanted to test that to see if this also changed about 20 million years ago. And the thing is that even though most Lamiaceae and Salvia have stigmas that are bilobed with a longer lower lobe and glabrous, this is what we find in most Calophaceae. They have a pubescence below the stigma, and they have a very long upper stigma lobe. This is very unusual for the whole order Lamiales, I dare to say. For sure, for family Lamiaceae, we did not find anything similar in the whole family. What are those structures doing? We don't know, but we've hypothesized that the upper stigma lobe helps the style anchor on the corolla. So if you see here this Wagner sage, you can see that stigmatic lobe anchoring on that corolla. And why we hypothesize that? Because then when the lever functions, then the style stays in place. The style does not move downward and cause intrafloral interference. So that's the hypothesis we have right now. If anybody wants to help test me or has another one as to why that stigma has that lobe, please reach out to me. With respect to the substigmatic brush, we usually find pollen there from the same flower, it appears to be. And here again, it's a hypothesis that we've come up with, that it is a type of secondary pollen presentation. And that as the lever keeps working, it doesn't matter if it's devoid of pollen at that point, it could keep shaking pollen from that brush onto a visitor. So this study by Elgazar noted this, and they actually drew it. And they said, you know, these salvias from Mexico to the south look distinctive because of the pubescence and shape, but we're not sure how to measure it. So we're just going to leave it at that. And so at this point, we were like, wow, let's revisit this with our morphometrics. Let's take the stigmas. Let's do the morphometrics of the stigmas. Let's estimate a morphospace. Here we have in the x-axis to the left, the stigmas with the very long upper stigma lobe, and to the right, the ones with the longer lower lobe. We're going to match that data, which is the trait that we wanted to test, to the phylogeny and ask, when did it change? Or did it change many times? And what we find is that about 20 million years ago, when hummingbird pollination evolved, the stigma changed the morphology. And through time, there were a few reversals. The taxa that reverted in this case are taxa that are hummingbird pollinated, but with exerted stamens and styles. There's been diversity in those hummingbird flowers also, so these may have lost the need to anchor on the corolla because they're exerted. So to summarize these results, we get this ancestral bee flower that has 5 characters: the curvature, the pubescence, the stigma, and barely fused stamens at the base. And it differs in those 5 characters from the hummingbird flowers, which have straight styles, straight stamens, pubescence, upper longer stigma lobe, and very few stamens. But the bee taxa that we estimate are derived from hummingbird pollinated ancestor share those 5 characters. Okay, so I found this result really fascinating because now we understand why we have these different types of bee flowers. Well, some of them are derived from a hummingbird pollinated ancestor, and the hummingbird likely selected for these traits that were then retained when they reverted to bee. And just to show you here to the left is the phylogeny of hummingbirds, and that node at the base, about 20 million years ago, these authors estimated hummingbirds were living in an AC area, which is a North America, Western North America, Central America. So it is very likely that they were living together and that indeed they started to pollinate salvia around that time. Okay, so the last story about this section is a little study we did to try to understand why the lever has evolved further complexity. So if you look at a mealy sage from the front, this is in subgenus Calophaceae, you'll see the lever in the back blocking access to the nectar, and then these structures that are barely sticking out. If I put the flower to the side, I remove the style, take out the stamen, you can see there the morphology, the filament, the connective, the anther, and in the middle, a squarey structure, which is what taxonomists have called sometimes teeth. And we observe bees visiting and getting the pollen on the back of their head. And as they're doing that, they inadvertently seem to be touching with their face those teeth. So we hypothesized that those teeth have evolved to help move that lever further back in the bee's head, where the pollen will be safe to be taken to another flower inadvertently by the bee. So we did an experiment to try to test that a little bit. If I take a tweezer and try to see if this flower levers normally, it does. But if I just touch the teeth from the outside, the part that's sticking out and not enter the corolla, they also lever. So we suggest that these teeth are helping move the lever, and this is important because the bees groom. And if they groom that pollen away, you won't reproduce. But if you can put that pollen in a safe spot in the back of the bee's head, you might be safe. And here's a video of a bee, how she grooms when she leaves the flower. She grooms. Here's another example. So plants that are able to reach farther in the back of the bee's head might do better, and that selects for the presence of those teeth. Now, those teeth, we then went ahead and updated the Himmelbau and Stiebel diagram. We added the taxa without a lever. We added some potential pollinators to certain sections. But what I want to focus here is to the bottom right. Our mealy sage is there in the middle, but above them are the hummingbird ones. They also have a tooth. That's what I mainly want to point out here. The tooth may have evolved similarly to the other changes that I just talked about of the stigma, style, and lever. Okay, so now moving on to the second way that salvia flowers move, I want to introduce you to what we're calling the petal lever mechanism in some California salvia. Now, California salvia are in subgenus Audibertia, and they are fascinating. And one reason that I find them fascinating is that a lot of them have stamens to the sides. Very unusual for salvia. Most of the 1,000 recognized salvia have stamens hidden in the upper corolla lip. But in subgenus Audibertia, we get about half of them doing these stamens to the sides, which these flowers have been called pleurotribic flowers in other groups. Pleuro, middle, tribi, rubbing pollen in the middle is the idea, instead of nototribi on top or sternotribi in the bottom. Okay, so this is really particular, but we still don't know if they lever. But what we first did was we tried in this paper to map those stamens to the size on the phylogeny. And what we find is that it is possible that the ancestor had that kind of flower. So it might be that it is very important in the evolutionary history of Audibertia to have evolved the stamens to the sides. But we cannot be totally certain at this time. It leaves us with the question, though, do they lever? We still don't understand if they lever. And there was a study on Salvia appiana in 2016 where the authors say all species of Audibertia lack the lever mechanism, except the most common, Salvia columbaria. Now, if I show you a picture of Salvia columbaria here in both flowers, you can see the connective is prolonged and the two theca are offset. So it seems reasonable to believe this species levers. But what about something like Salvia appiana? And as we studied the pleurotribi, I noticed here the stamens are too high compared to the stigma. So if a bee visits this flower and gets pollen near its head and then goes to another flower, it's not going to touch the stigma that's near its abdomen. So what's going on? Well, what if we actually press that petal, pressure it or press it with a tweezer? The flower moves down. Okay, so one way to overcome that distance is with a lever mechanism that is a little different. It's different because the stamen is adnate to the petal, and it is weight on the petal that causes the stamens to lever with it, not with a staminal lever mechanism. And this is a bee I watched at San Francisco Botanical Garden, and you'll see when it visits this next flower, it causes the stamens to move downwards. So they do lever, some of them, and we still don't know the whole story. Now, I want to point out that I did find a reference from 1970 by Frank Visco and Brian Capone, and they actually say that the two stamens in thistle sage produce a lever mechanism similar to that of Salvia patens. As the lower lip of the corolla in Salvia cardoacea is depressed by a flower visitor's weight, the anthers are spread to the lateral portions of the flower, allowing access to the nectar for the bee and bringing the anthers in contact with the bee's body. So there actually was a reference, and I believe that because Cardoacea looks so similar to Apiana in the morphology of the stamens and style, that it is actually a petal lever, not what Salvia patens have, which is a regular staminal lever. Now, interestingly, in the phylogeny, Cardoacea and Apiana are distantly related. So it is possible that the petal lever mechanism did, in fact, evolve multiple times within this group. Lastly, I wanted to share a third way that salvia flowers move and a clade that appears to have lost it. And so protandry is very common in Lamiales. If you have a nectar reward, you can separate sex and time, be male first and female second. That is protandry, and always provide nectar as a reward. And here you can see American germander. The anthers have pollen, but the stigma is closed. Through time, stamens move out of the way and the stigma opens. Here's another germander. Same thing, protandry, evident protandry. We get to the Himalayan mint shrub. It also has the protandry, and when we get to salvia, here's rosemary moving from male to female phase. Same thing, hummingbird sage. And species of two other subgenera where you can see the flower to the left can be visited, but the stigma is closed, the lever will work, and then the stigma opens. So what happens when we get to our famous Salvia subgenus Calosfaci? Well, as the flower develops, the stigma lobe gets anchored. The style is straight, so it's not going to bend downwards. And so it appears that this group has actually lost protandry. And there is there's a study on pollination that noted in one species of this group that it wasn't protandrous. Now we haven't tested many, so that's what we're working on. And the way we're doing that is by using hydrogen peroxide on flowers of different ages and looking how the peroxide reacts to the peroxidases in the stigma when it's receptive. Here to the left, you can see a stigma bubbling all throughout, and that's the ancestral state. And to the right, you can see a subgenus Calosfaci, and the bubbling is concentrated to the apex of the lower stigma lobe. Here's a close-up of Wagner sage, which shows it really nicely. The receptivity is very restricted to that part. So we end up with protandry being predominant. We have the outgroup has protandry, but then Calosfaci appears to have lost it. Although we don't know why, there are suggestions that hummingbird pollination may have selected for the loss of protandry because hummingbirds forage differently. They don't forage upward inflorescences. And it has been suggested that foraging upwards, like bees and other insects do, might select for protandry by having older flowers in the bottom be female as they're older, and the top ones being male as the bee comes, visits the lowermost, touches the stigma, and then picks up as it visits the uppermost flowers. And so we're still studying this, but I really wanted to share it because it really is suggesting more evidence that hummingbirds may have changed these Calosfaci strongly. So with that, I want to share that we are still working on more things. We're expanding the next-gen sequencing phylogeny. We have about 300 samples now. We're also trying to then determine and map finally how many times the lever may have evolved. And we're trying to also look at the evolution of flower color because we think that color is going to be very important. To do that, we also use our plates. We can extract RBG scores and get the main score for each species, and then look at shifts in color, for example. And we're not only looking at flowers. I do want to share, we've been starting to look at the leaves, leaf shape and leaf type. We've generated a data set and a morphospace. We've looked at shifts in leaf shape and finally in leaf type. And the spoiler here is that compound leaves have evolved more than one time in the genus Salvia, which was really surprising to me. Now, for conclusions, I want to say that we find major changes in flower morphology that occurred in America about 20 million years ago. And we think that hummingbirds, and our data suggests that hummingbirds drove floral evolution in New World salvia, resulting in a different type of bee flower, the stigma mohawk and upper long lobe bee flower. Now, we find that some California salvia also lever, likely in more than one way, and they also have a different type of bee flower, many of them. That is the pleurotribic bee flower. Subgenus Calophaceae may have lost protandry due to the early evolution of hummingbird pollination. And lastly, pollination syndromes are useful, and adding morphology helps gain deeper insights into flower evolution. And with that, I want to thank the California Botanical Society for inviting me, my collaborators, also Holly Forbes, who helped us be able to sample at UC Garden when we first started, and also all my collaborators and volunteers at Cal Academy, and my great professor, Bob Patterson, who I know is watching. Thank you, Bob. Thanks.
That was terrific.
Continue to share.
Yeah, we have questions in the chat.
Let me see if I should stop sharing here. Okay. Then I go to the chat.
Yeah, funny, I'm not seeing it. We have hands up as well. Peter, Albert, do you want to unmute?
Well, I'm going to ask, I think Andy has a question about the uncertainty margins and the dating of your phylogeny.
That was the question. Is you put up this phylogeny and you said that 20 million years ago changes happened. There was the switching to hummingbird pollination, and there was the colonization of the New World. But I am wondering what the uncertainty margins are, because tying that to geologic events is potentially interesting, depending on what the distances across the barriers to dispersal were at different points in time.
Yeah, Andy, thank you for that question. I'm not an expert in salvia, so I definitely read up a little bit on that because I want to understand better. And I know we use there's a couple of fossil pollen grains that were used. That was part of the way we calibrated the tree. But I do want to mention that the older dispersal from the old world, we find that it was likely happened through the Bering Land Bridge, which was suitable for exchange.
Came across Beringia. Okay, that's what I was wondering.
Yeah, that's what we estimate for that one. But I don't remember the margins exactly. But yeah, that's the Beringia is the hypothesis for that one.
Makes me wonder how they got into South America then, because the Isthmus of Panama is younger than that.
You know, great question. I mean, some of these have been actually found to have glandular hairs on the calyx. There's a paper by Scott Zona on dispersal of salvia that, does raise the possibility there's different ways they could have crossed.
Peter?
Yes, what an attractive subject and such interesting work. So I was wondering, does the hummingbird morphology make bee pollination less effective? or um, are the bees equally effective with either the bee morphology and the hummingbird morphology?
Peter, that is a great question, and that is sort of right now what we're thinking a lot about. You know, hummingbirds are great pollinators. Why switch? And I mean, one of the reasons maybe there's competition for such great pollinators like salvia and like hummingbirds, and if you can switch to bee, then you might be okay. So competition for the hummingbirds was one of them. But also, it appears, Peter, that once you were derived from that hummingbird ancestor, it appears that having that stigma clinging there in some way affected flower morphology, and then you get these bee flowers with very long lower corolla lips. And so it seems as though being derived from hummingbird may have actually helped some of these bee lineages be a better bee flower, if that makes sense.
Yeah, okay, thanks.
You're welcome. But if people have ideas, do send me a message. Why would we switch back to bee many times? Another reason actually has to do maybe with diversification of the bumblebees. There's this really interesting study by Dr. Heather Hines on the biogeography of bumblebees, which shows that bumblebees really diversified in America maybe 5 to 10 million years ago, a lot of spurts of bumblebees. So I wonder if maybe some salvias took advantage of those bumblebees.
Anyone else? You can unmute yourself and just ask your question. Glenn?
I was wondering, do you get pollination by other bee families besides Apidae? I think all the examples you showed were Apidae, but there are other bee families as well. Do they participate with Salvia?
Great question. Glenn, I don't have I do believe so. You know, I can't tell you exactly the genera, but there's at least some papers, and I believe there are definitely other genera involved, other families, sorry, but I'm gonna owe you that because Apidae is most of the bees these days, right?
Apidae is the largest family.
Yeah.
There's some others that are quite large as well.
Yeah. So I think I've seen like a megachilid bee visiting a leafcutter bees, and I wouldn't be surprised that other bees also visit salvia. There's some studies in the Mediterranean. I just don't want to guess exactly, you know. So I'm not sure, Glenn, but I would think so. I don't see why other bees would not visit the salvias
Thank you.
for nectar. Yeah. I'll think about those bees, but
All right. Anyone else?
Well, can I ask another question?
Yes, you can.
So in the picture you had of the bee flower becoming the hummingbird flower, becoming a new kind of bee flower,
yeah.
You start out white and then you go to red with the hummingbirds, and then you go to blue with the bee the second time around. But you didn't mention flower color, the role of flower color in all of this. Does it play a role?
So great question, Peter. That example, I didn't have a blue flower. I think it was mainly because I had a really nice. I think the species that I showed, I had a good picture showing the barely fused stamens. But there are blue flowers for sure in the ancestral morphology. So yes, color is probably very important. Because if you ask me, then what are the differences between the bird and the bee from bird? What's left? You know, and one of them is color. And so, yes, I think color is very important. And we do have a data set, but we want to make it a little larger. And so together, you know, we want to test the hypothesis that red evolved about 20 million years ago and then was lost subsequently in some of these blue flowers or changed. So that is our next step, Peter. It's just somewhat challenging to quantify color, and I didn't want to do it in a discrete way because it's impossible and I wouldn't feel good about it. There's all kinds of hues. So there's now there's ways to now use the images to try to get RBG colors, RBG scale values, and do it that way.
I'll look forward to it. Thanks.
You're welcome.
Good.
So I see a question by Esteban. Does humidity affect Salvia's evolutionary morphology? You know, I don't know, but I do want to say that because of the lever mechanism, stamens in Salvia can be held in that upper lip and be very protected against the rain or larcenists. There's those other bees, the trigona bees, that like to steal pollen so much. And so it's possible that gallia and the lever having the stamens there is protecting them from some of that humidity. But I'm not sure in general what humidity does to salvia flowers.
Glenn,
Yes,
do you have
Glenn.
another question?
There's it was interesting that you've tracked a movement from the old world into the new world, but more diversity here. Of course, one thing that we have here that they don't have where the genus originated are hummingbirds, but I've also, my understanding is that bee diversity is also higher in North America than many other places on Earth too. Mm-hmm. And it would be interesting whether the bee diversity here has helped salvia evolution.
Yeah. Yeah, it's interesting because as far as I remember, the most diversity is in the deserts of the bees.
That's
and
correct.
That's not where the most diverse clades of salvia are. Right? So I still think you're up to something, and that the bees are helping them diversify. But with respect to what I remember is that in North America and in the West, the deserts select for a lot of oligolectic bees that specialize on plants, and there's a lot of them. But yeah, maybe the bumblebee radiations fall into sort of what you're talking about too. But
Jennifer, did you have a question?
And also out of the deserts into the Mediterranean climate area as well. So it's you're certainly is very diverse in the deserts, but basically the Mediterranean climate has considerable diversity as well.
Glenn, I wanted to ask you, what do you think salvias could be competing with in the Mediterranean climate that might have forced them to evolve stamens to the sides? This is something I'm stuck with right now, Why did they evolve those stamens to the sides? My guess is that they avoided competition with somebody that was really good at putting them on the head of the bee. That's so I'm brainstorming with all of you because I don't have the answers. I'm just contributing to the discussion, but yeah, I'm puzzled about that.
Jennifer?
Oh, thanks. I had a question about so with all of this phylogenetic work is super fascinating, but I'm wondering if you have any evidence, like anecdotal or otherwise, that some of these traits, like lever mechanisms or even flower color, like any of these traits might vary within species across some of the strong environmental gradients that we have in California and some of the other places you're talking about.
Within species, Jennifer, right?
Mm-hmm.
Yeah. So I'm sure there's variation within species, but for example, some of these traits seem to be pretty conserved, the ones that we saw today. So I'm sure there is variation, and I know there is color-wise in some of them, but in my limited experience as a systematist of salvia, I gotta say, I haven't heard, for example, a lot of species complexes. I don't hear a lot of people talking about species complexes and what is this salvia. in California, it doesn't, they do hybridize, but for the most part, it seems like people can identify them. And so I do think that that is a great step and is a difficult one, but we wanted to also try to get that, Jennifer, with the plates. Because when we go to a population, we can do plates of each population, and we don't have to sort of like just tell you they're this way. We can give you the spread, you can analyze it, we can analyze it, but to try to get at that question, a better idea of the variation. But we haven't gotten there. We've done 400 plates, but most of them are different species at this moment. But that was the goal. So I owe you more about intraspecific variation. Mm-hmm.
Yeah, your plates were looked so cool. That was exactly what made me think about it, is like how much variation do we have within the species too? Yeah. Awesome.
And as soon as we find variation, it's going to be exciting because there's probably going to be stuff to try to understand there. Yeah. Oh, I think there was another question here by Jasmine. Is there a higher rate of nectar robbery by bees in the salvia suited toward hummingbirds? You know, Jasmine, I'm not sure because I don't, the nectar robbing that I've seen in salvia, here I've seen a hummingbird is the one that I've seen robbing nectar. So I'm sure bumblebees and others steal nectar in salvia, but the rates of robbing in salvia, you know, that's a great question. And I don't know that we know, you know. I've barely seen robbing documented, so that's definitely a nice study for someone. I will say that somebody has studied nectar robbing and included a salvia, but that's it. Not a lot of comparisons at that level.
Yeah, I've definitely have seen nectar robbing done by carpenter bees on some red salvias. So I was very curious because I know bees generally can't see red, but they still go to red flowers because of other factors. So I was wondering what if maybe that's why they're going to those ones and nectar robbing.
Very cool.
Yeah, the nectar robbing is also tricky because salvias mostly have levers. It's not just any bee that can go there and steal nectar. The carpenter bee can make a hole at the bottom. The ones that can do that can steal, but going through the tube is going to be harder for a robber.
Okay, I think it's probably time to wrap it up. Let's thank Ricardo again for a spectacular talk. Thank you.
Thank you, everyone. Thanks so much. I really hope you enjoyed it.